Hello, my name is Carl, and in this video I'm going to talk about negative inducible gene regulation in prokaryotes. For this video, we're going to explain the concept through an example. We're going to use a fictional operon called the Ricecrispyase operon that encodes several genes required for the metabolism of our substrate, but the gene that we're going to be looking at specifically is for Ricecrispyase. Our operon is composed of our promoter, our structural genes, and our operator. Our gene of interest produces an enzyme called Ricecrispyase. Additionally, we'll be looking at the gene that produces our repressor protein. It is composed of the promoter and then the sequence that produces the repressing protein. The whole point of this process is to regulate the catalysis of a reaction using this enzyme that breaks down our substrate, which is Mintose. First, let's look at how this gene expression works when our substrate is not present. RNA polymerase bonds to the promoter on the sequence that produces our repressor protein and transcribes and then is translated into our repressor protein. Our repressor protein binds to the operator for the Ricecrispyase operon, and then RNA polymerase is unable to bind to and transcribe our gene of interest. Consequently, the Ricecrispyase enzyme is not produced. Now let's look at what happens to gene expression when our substrate is present. Again, RNA polymerase transcribes the gene that produces our repressor protein and it is translated into our repressor protein. But now a molecule of our substrate actually binds to the repressor protein and makes it unable to bind to the operator of our operon. Now RNA polymerase can bind to and transcribe our gene of interest. Ricecrispyase is produced and it binds to and then catalyzes the reaction that breaks down our substrate, Mintose. So for a moment, let's think about why a process like this might evolve in the first place. Well, when our substrate isn't present, we don't actually need the Ricecrispyase at all. And so by not transcribing it, we save energy and resources until the substrate is present. Then we produce the enzyme that is needed to break it down. This type of regulatory system might save an organism energy and resources over the long run. Well, that's it for this video. Thank you for watching. Hopefully now you understand this regulatory process a little bit better.